People have been uncomfortable with the strangeness of quantum mechanics for around a century. It is not surprising that people would be similarly uncomfortable with the idea of evolution involving a degree of purpose. I have gathered some videos that each seem to hold a piece of the puzzle pointing to the possibility of this purpose. This has been a theoretical endeavor on my part, and further research in labs and such should be able to support or show gaps in my conclusions. Some of these videos helped me determine whether my idea of the environment influencing evolution could be possible back when I was writing my book, Purposeful Evolution. It is such a counterintuitive idea, and I did not actually expect to find a viable mechanism for this process. However, I think that I did actually find a viable mechanism combining the reactiveness within a cell, information being carried by biophotons, stress influencing the emittance of these biophotons, all living organisms containing these biophotons, quantum mechanics being observed at the cellular level including its possible ability to affect the emergence of mutations, the apparent reactiveness between the emergence of mutations and the organism's environment, and of course natural selection. I hope I have captured these various ideas using the following videos, and the combination of these ideas is made as evident for you as it was for me years ago when I was trying to figure this all out. This video does a good job of introducing relevant terms, and it seems like a good idea to start at the foundation of the topic. I have chosen to let the clips finish before sharing my input on the ideas in each video. I would rather not keep stopping the videos and disrupting the flow. Sorry if this way of doing it bothers you.
Quantum biology asks a very simple question. Does quantum mechanics, that weird and wonderful and powerful theory of the subatomic world, of atoms and molecules that underpins so much of modern physics and chemistry, also play a role inside the living cell? In other words, are there processes, mechanisms, phenomena in living organisms that can only be explained with a helping hand from quantum mechanics? Now, quantum biology isn't new. It's been around since the early 1930s. But it's only in the last decade or so that careful experiments in biochemistry labs using spectroscopy have shown very clear evidence, firm evidence, that there are certain specific mechanisms that require quantum mechanics to explain them. Quantum mechanics was developed in the 1920s. It is a set of beautiful and powerful mathematical rules and ideas that explain the world of the very small. And it's a world that's very different from our everyday world made up of trillions of atoms. It's a world built on probability and chance. It's a fuzzy world. It's a world of phantoms where particles can also behave like spread out waves. Quantum biology is about looking for the non-trivial, the counterintuitive ideas in quantum mechanics, and to see if they do indeed play an important role in describing the processes of life. Particles can multitask. They can be in two places at once. They can do more than one thing at the same time. Particles can behave like spread out waves. It's almost like magic. Physicists and chemists have had nearly a century of trying to get used to this weirdness. I don't blame the biologists for not having to or wanting to learn quantum mechanics. You see, this weirdness is very delicate, and we physicists work very hard to maintain it in our labs. We sort of cool our system down to near absolute zero. We carry out our experiments in vacuums. We try and isolate it from any external disturbance. That's very different from the warm, messy, noisy environment of a living cell. Biology itself, and you think of molecular biology, seems to have done very well in describing all the processes of life in terms of chemistry, chemical reactions. And these are reductionist, deterministic chemical reactions, showing that essentially life is made of the same stuff as everything else. And if we can forget about quantum mechanics in the macro world, then we should be able to forget about it in biology as well. Well, one man begged to differ with this idea. Erwin Schrödinger, he of Schrödinger's cat fame. He's an Austrian physicist. He was one of the founders of quantum mechanics in the 1920s. In 1944, he wrote a book called What is Life? It was tremendously influential. It influenced Francis Crick and James Watson, the discoverers of the double helix structure of DNA. To paraphrase a description in the book, he says, at the molecular level, living organisms have a certain order, a structure to them that's very different from the random thermodynamic jostling of atoms and molecules in inanimate matter of the same complexity. In fact, living matter seems to behave in its order, in its structure, just like inanimate matter, cooled down to near absolute zero, where quantum effects play a very important role. There's something special about the structure, the order inside a living cell. So Schrödinger speculated that maybe quantum mechanics plays a role in life. This is one of the best known phenomena in the quantum world, quantum tunneling. The box on the left shows the wave-like spread out distribution of a quantum entity, a particle, like an electron, which is not a little ball bouncing off a wall. It's a wave that has a certain probability of being able to permeate through a solid wall, like a phantom leaking through to the other side. You can see a faint smudge of light in the right-hand box. Quantum tunneling suggests that a particle can hit an impenetrable barrier and yet somehow, as though by magic, disappear from one side and reappear on the other. The nicest way of explaining it is if you want to throw a ball over a wall, you have to give it enough energy to get over the top of the wall. In the quantum world, you don't have to throw it over the wall. You can throw it at the wall, and there's a certain non-zero probability that it'll disappear on your side and reappear on the other. This isn't speculation, by the way. This is, we're happy. Well, sorry, happy is not the right word. We are familiar with this. Back in the 70s and 80s, it was discovered that quantum tunneling also takes place inside living cells. Enzymes, those workhorses of life, the catalysts of chemical reactions. Enzymes are biomolecules that speed up chemical reactions in living cells by many, many orders of magnitude. And it's always been a mystery how they do this. Well, it was discovered that one of the tricks that enzymes have evolved to make use of is by transferring subatomic particles like electrons and indeed protons from one part of a molecule to another via quantum tunneling. It's efficient, it's fast, it can disappear, a proton can disappear from one place and reappear on the other. Enzymes help this take place. This is research that's been carried out back in the 80s, particularly by a group in Berkeley, Judith Klinman. Other groups in the UK have now also confirmed that enzymes really do this. And one question we asked is whether quantum tunneling plays a role in mutations in DNA. Again, this is not a new idea. It goes all the way back to the early 60s. So the two strands of DNA, the double helix structure, are held together by rungs. It's like a twisted ladder. And those rungs of the ladder are hydrogen bonds, protons that act as the glue between the two strands. So if you zoom in, what they're doing is holding these large molecules, nucleotides, together. Zoom in a bit more. So this is a computer simulation. The two white balls in the middle are protons, and you can see that it's a double hydrogen bond. One prefers to sit on one side and the other on the other side of the strand. So the two strands are the vertical lines going down, which you can't see. It can happen that these two protons can hop over, watch the two white balls, they can jump over to the other side. If the two strands then separate, two strands of DNA separate, leading to the process of replication, and the two protons are in the wrong positions, this can lead to a mutation. This has been known for half a century. The question is, how likely can they do that? And if they do, how do they do it? Early indications suggest that quantum tunneling can play a role here. We still don't know yet how important it is. This is still an open question. It's speculative, but it's one of those questions that is so important that if quantum mechanics plays a role in mutations, surely this must have big implications to understand certain types of mutations. Some years ago, the world of science was shocked when a paper was published showing experimental evidence that quantum coherence takes place inside bacteria carrying out photosynthesis. The idea is that the photon, the particle of light, the sunlight, the quantum of light, captured by a chlorophyll molecule, is then delivered to what's called the reaction center, where it can be turned into chemical energy. And in getting there, it doesn't just follow one route, it follows multiple pathways at once to optimize the most efficient way of reaching the reaction center without dissipating as waste heat. Quantum coherence taking place inside a living cell. A remarkable idea, and yet evidence is growing almost weekly with new papers coming out confirming that this does indeed take place.
The ability for quantum mechanics to work as well in cells as particles do at near absolute zero is critical to my proposed mechanism. Another very important aspect is quantum tunneling because this mechanism involves the transference of photons between cells. Also, photons would be unable to leave an organism's cells and enter the cells of another organism if quantum tunneling was interrupted in biological systems. Photons acting like waves helps the transference of information carried by these photons because organisms are sometimes spread out. Scientists already determined quantum tunneling might affect mutations, which is a plus because that would make my proposed mechanism more likely to be correct. The coherence involved with photosynthesis is important because photons involved in this mechanism have a high level of coherence, which is explored in a later video. This video explores some surprising facts about a brainless animal. Cells involved with genetic changes are brainless, so this is an important piece of the puzzle.
When researchers looked at trichoplax under a microscope, they were shocked to see its millions of cells engaged in complex activity. Trichoplax was able to coordinate its movement, strategize how to find food, and even reproduce by ripping itself in two. But how can trichoplax exhibit these complex behaviors without a brain to call the shots?
Traditionally, when you think about intelligence, you think of the brain. Trichoplax stretches that intuition, stretches your imagination to ask, how intelligent can a collection of cells with no neurons, no muscles, actually be? How much intelligence can a mechanical collective embody?
To answer these questions, the researchers tried to understand the simple mechanics of the trichoplax, starting with the tiny waving filaments called cilia.
Cilia I have often described as the atom of motility. For the last 50 years of the work in ciliary biology and ciliary biophysics, we have thought of cilia as an actuator that enables swimming.
Typically in organisms with nerves and muscles, cilia help move fluids within the body. But when the Prakash lab studied the cilia on the underside of the trichoplax, they discovered something unexpected.
I explicitly remember a moment when we started imaging the organism and literally watched the cilia. What we observed was a walking gait, and the realization was that the organism does not swim, it actually walks.
But just how these individual cilia cells coordinated their movement remained a mystery.
We started to characterize our work as asking the question, how do you walk with a million legs? How can many of these cilia work together?
When the researchers looked at all the ciliated cells at once, they saw something they never thought was possible. The cilia synchronized their movement like a flock of birds.
To literally be able to watch this kind of what we end up describing as a flocking dynamics was surreal because you could see it. And, when I see bird flocks, I can imagine, there is a controller, there is a brain in there that's making these sets of decisions. Here we are talking about individual flocks of these cilia that are seemingly just connected by springs and twirling in remarkably organic ways.
Individual cells, the researchers found, could self-assemble into collective systems capable of a wide range of behaviors, including some that allowed them to be responsive to their environment. In other words, basic mechanics could give rise to the kind of incredibly complex behavior we normally expect from brains.
The observation of Trichoplax's ciliary flocking really brought to the fore for us this idea that mechanical systems could somehow be intelligent. They could somehow seek out food. They could somehow come to consensus across many cells. And they could somehow find ways to keep an organism alive as a whole instead of just its individual parts. One of the implications of our work is that it really has pulled us in new directions as we think about computation and intelligence in physical matter.
The reactivity this animal has to its environment, even though it doesn't have a brain, is the important factor here. The cell can't start the seemingly stochastic process of developing beneficial mutations based on environmental factors such as stressors if this type of reactivity is not possible at the cellular level. This video does a good job of introducing biophotons, which are a main factor involved in my proposed mechanism.
I will show you our results in which we captured light from the living cells. And from now we conclude that every cell, either it is a plant, either it is a microorganism, or any living being, all of us, emits some kind of photons. And these photons are just not useless. It has important functions to do. I will demonstrate in the results here. So I came from India to work on this field. The inspiration for the study came from this person. In 1923, what he did, he found out that when two onion tip cells were kept together to each other and it was separated optically, that means there was a barrier between the two onion tips, and there was no chemical transfer, no gaseous transfer, but there was an influence of one onion tip on the other tip. That means the growth of one was influenced by the growth of the other. So what actually is the intensity of the emission which we emit? It is just a few photons per second per square centimeter. So the difficult thing here was the detection, how to detect this. So what we did is, before we start the measurement, we need something, some chamber, which will be completely dark. We tried to construct this. What we did, we constructed 3 layers of dark chambers, and the final, the innermost one, was considered to, and we measured there, that there are no photons going inside. There is no source of photons inside that room. Now, the next thing was, actually, it looks something like this, if you look inside the room. So after that was, this next step was to detect it. So we use the photoelectric effect. What happens in this effect is, if there are some photons, when you direct this to some photocathode, it ejects some electrons from this photocathode. And in course of time, with this series of photocathode, it multiplies the electrons, and finally it is detectable. The upper one is the picture of the hand taken with a normal photograph, and the below 2 images are the bioimages. We call it biophoton images or ultra-weak photon emission images. So these are the real photons which are coming out of your body. This was measured from the hands, both the sides of the hand, and next was this. So this is the photons which each of us emits. This was measured for 30 minutes. So the top 2 images, the first one is just a picture. The second one is the spontaneous one. By spontaneous, I mean which is spontaneously generated without any excitation in normal. And the below 2 images is, we treated our hands with some kind of stress. We applied some very low concentration of some chemicals which are doing some oxidation and reduction. So the image was enhanced in these 2 cases. So the conclusion from this was, the emission comes from our biomolecules in the body. Now, a parallel group who is working in Japan recently produced these images, and what he is saying is, from this picture, the picture on the right is the back of a mice. The mice was induced with basal cell carcinoma, some kind of cancer, and he observed that when there is a cancer, the portion which was induced with the cancer emits a high level of photons. So that means the metabolic activities, because in cancer cells there is a high metabolic activities, there are rapid cell divisions. So this was the reason for high emission. Okay, that means we can measure some kind of stress from this. We found out that the emission is in the visible region of the spectrum. That means starting from the blue till the red end of the spectrum. And also, to our surprise, it was that when there is a healthy living cell, it emits blue photons, while when the cells are diseased, they emit in the red region of the spectrum. So the next experiment was to understand why these photons evolved in nature. With these experiments we are doing, it is in the preliminary stage, we found out that when we kept different microorganisms in close contact, one was metabolically active, the other one was metabolically less active. That means one was having high density, dividing rapidly, and the other one just a few cells dividing slowly. We monitored it by various techniques, and we find that when we brought in a close contact, closed the cover, didn't allow anything to pass, nor volatile compounds, no chemical transfer. And the surprise was that the population, the small population, was influenced by the division of the high population. And in few hours, the rate of division in the other population was increased drastically as compared to the control. So this can be an explanation why millions of reactions which are going inside our body are so well controlled. Maybe the signaling in our body for the reactions mechanism is controlled by photons somehow.
The reactivity of cells to possible stressors in the environment would be greatly hindered if only certain groups such as animals emitted these specialized photons. The commonness of these photons means they can potentially help any organism adapt to their environment through information gained from biophotons. It is not surprising that biophotons have not been considered in the emergence of new mutations. For one, questioning the idea of mostly random mutations as featured in Darwinian evolution is unlikely for most people since the idea has persisted for over 160 years. The other reason is biophotons are quite difficult to detect even for people suspecting their existence. This video also introduces the change in how many biophotons are emitted from biological systems when a stressor is present. The addition of stress to the organism increases the number of biophotons it emits. This is useful in terms of biophotons helping the emergence of beneficial mutations because other organisms in the environment are more likely to come into contact with these photons. This could cause other organisms in the environment to undergo the seemingly stochastic process of developing beneficial mutations. This would hopefully increase the odds of a beneficial mutation emerging within the population. Then natural selection can test this new mutation and spread it to others in the population through reproductive success. This video contains arguably the most important piece of the puzzle for how biophotons can affect the mutations of organisms based on stressors experienced within an environment.
I interviewed German biophysicist Professor Fritz Albert Popp. Now, he's known for his work exploring biophotons, and now biophotons are ultraviolet and ultra-weak light that emanate from all living systems, including humans. Now, the biophotons in our cells function as regulators or carriers of information. So biophoton emissions, they provide an ideal communication system for the transfer of information to many cells across our body. He also examined the effects of stress. When we're stressed, the rate of light emissions goes up. So this begs the question, what actually creates a photon emission in the cell? And as the professor will explain, there needs to be an order, a coherence, and timing is everything.
It is, the photons have to be very coherent. They cannot be ordinary photons. The coherence time of an artificial photon is in the order of, say, a nanosecond. The coherence time of the photons of a laser is about a tenth of a second. But you have to consider coherence times which are even much longer, or the degree of coherence should be much higher. By the way, last spring is now this spring in Berkeley in California, they showed evidence that the quantum coherence in photosynthesis is much higher than expected. So the efficiency of photosynthesis is much higher than they expect. There is almost no entropy production with that because this coherence is rather high. It has it plays a big role, a very important role, for instance, for the heat, for the warming of the Earth. And not only there, but in all biological systems. You have this perfect efficiency of the different reactions, which is only possible with a more or less a perfect quantum coherence of all the things which happen. So these photons in a biological system have a very, very high degree of coherence. And we showed evidence in the last years of that. You can show it by means of the possibility of statistics or photo count statistics. The photo count statistic can give a proof of the high degree of coherence. And we showed evidence already. We took about 10 years in order to show that, and to measure the degree of coherence of biophotons. So we are convinced now, we have to know that coherence is not only important for the visibility and for the different functions of information transfer. It is also important for the distance over which you can transfer information. At rather high degrees of coherence, which are technically not possible, but in our biological systems they are possible. You have even to consider that the information can get out of the body over a longer distance and can be transferred and can be controlled and regulated over many say kilometers of distance. But it is very difficult because with increasing coherence in this range, the matter becomes transparent. So over long distances, you have the possibility to transfer information in terms of resonance principles. If you have a complicated antenna here and a complicated antenna here, which can transfer information by resonance, you can do it over many kilometers of distance. All these different kinds of information become possible by simply by coherence, by a high degree of coherence. And so the information gets completely different ways as we are commonly used of.
And so I asked the professor whether this is like saying that the information exists anyway, but two people can access it at the same time. Like information that is recorded in a reality, but the patient and the therapist are just accessing it simultaneously.
For instance, we speak of zeitgeist sometimes. So many people think the same things. And it is a tuning into the surroundings and the environment. And the information character must not be personal. It may have many different roles.
The high coherence of biophotons is of critical importance to my proposed mechanism. These information-carrying photons would be of no use if they didn't last long enough to spread information to others in the vicinity. Cells changed based on information about environmental stressors could use biophotons to spread the useful information to other cells throughout the organism. I think this mechanism could also account for epigenetic changes in DNA. An organism would not benefit from maintaining a novel system that helps with a stressor in the environment if that stressor is temporary. Epigenetic changes allow organisms to have beneficial mutations without their descendants needing to maintain genetic changes unless the environmental stressor is still a significant problem after X amount of time or over multiple generations. An important feature in nature showed me the likelihood of mutations emerging through the spread of biophotons rather than only happening within the organism experiencing the stressor. That is the evolutionary arms race between organisms utilizing venom and resistance of their prey to this exact venom. It is highly unlikely for a victim to survive long enough to not only develop the appropriate mutation but also propagate this beneficial mutation throughout the population. It is most likely difficult to have reproductive success when you are dead from the venom you need higher resistance against. This video helps show the difference between the typical view of evolution and evolution caused by my proposed mechanism.
One of the things that a lot of people assume when they first learn about evolution is that mutations must be maximally random. You'll hear people say that there's random mutations and then there's natural selection. Well, that must mean that mutations are maximally random. Another thought is that random mutations are naturally biased due to physical constraints. DNA is a physical structure. It lives in a physical world. The cell is a physical structure. Many biologists have pointed out there are actual sequences of DNA that are more stable than other sequences of DNA and less stable and so on. That's going to cause biases in mutation rates across a stretch of DNA. And there's a bunch of other physical constraints that affect mutations. Some people have built on top of that argument, and they say that, genomes could evolve to exploit these natural mutation biases and then optimize the process of evolution. I'm going to focus on the first claim here that mutations are maximally random. We will also touch on these other two. Mutations are naturally biased due to physical constraints, and then the idea that natural selection could actually play with this. The other thing that we would expect if mutations truly were maximally random is we would expect that the mutation rate affecting a genome would be completely random, no rhyme or reason to it. In reality, what we see is that mutation rates increase when cells are stressed and decrease when cells are not stressed. This is really interesting and has caused a huge debate. Early on, a lot of people thought that maybe every time we see this, what we're seeing is we're seeing an evolved ability here. So if a cell is stressed out, that must mean that the cell is not fit for its environment. So it would make sense if that cell decided, oh, I should evolve faster now, and it would increase its mutation rate. So a lot of people thought that every time we see this, we're seeing that, the cell is trying to control its own evolution. Well, to assume that that's always what's happening here is obviously pretty silly, because when cells are stressed out, they simply can't perform well at anything, including maintaining their own DNA. But because this phenomenon naturally exists, of course, it is possible that natural selection could enhance this. And you actually could get cells that actively induce mutations when they're not doing well. They actively induce mutations because in the past, doing this made it so that they could sometimes achieve mutational rescue. Cell is a physical environment. It's a chaotic environment. Certain stretches of DNA are going to be safer than other stretches of DNA just because of the way that they're packaged inside of the cell. There are sections of the genome that are just more exposed to danger than other sections of the genome, right? This is just inevitable. This is an inevitable consequence of being a physical thing in the physical world. The question is, has natural selection exploited this fact? Is there a way in which genes that are really important could end up over evolutionary time residing in really secure areas of the genome, while genes or just random stretches of DNA that are not very important would end up taking up the more dangerous areas of the genome? Are there like kings and pawns within the genome? It turns out that there are mechanisms that could cause this to happen. There are certain types of mutations called translocations and transpositions that will shuffle where things are located in the genome, where certain stretches of DNA reside, because those mutations are constantly happening. If you had strong enough selection pressures, you could get natural selection to favor individuals that have their most important genes located and their most secure spots.
In John Perry's spectrum of thought, my proposed mechanism would fall under the third option, which says genomes can evolve to exploit natural mutation biases and optimize the process of evolution. He is understandably skeptical of this being the only mode of evolution. I agree, because there are plenty of examples of deleterious mutations. These mutations would not be possible if genetic changes were only caused by a purposeful process. The existence of random mutations has no impact on the likelihood of some mutations being influenced by stressors in an organism's environment. I enjoyed this video because it captures John Perry's reaction to the possibility of mutations being influenced by factors in an organism's environment.
If mutations are not fully random, does that mean that they happen, quote, on purpose? And if they do happen on purpose, what exactly does that even mean? People who haven't read the actual paper, but they just read the headlines, a lot of them are thinking, well, maybe cells have some sort of a way to reach inside themselves and cause specific mutations when needed. And if that's true, and if that's the norm, if that's how mutations normally happen, that really is in pretty big violation of what we thought we understood about evolution. Darwin presented evidence that heritable variation plus cumulative selection gives rise to adaptive evolution. So he did not believe that these variations or these mutations were purposeful. Natural selection can do nothing until favorable variations chance to occur. That's the word that he liked to use. He actually never used the phrase random mutation. He talked about chance variations. If we fast forward to the 1950s, we hear this from Dobzhansky. He was one of the major founders of what's called the modern synthesis, the merger between Darwinian evolution and Mendelian genetics. This is what he said about mutation. An ideal situation would be if the organism were to respond to the challenge of the changing environment by producing only beneficial mutations where and when needed. But nature has not been kind enough to endow creations with such a providential ability. G.G. Simpson, one of his comrades, another person who's considered to be a founding father of the modern synthesis. He was responding to people's questions about what random means in evolution. What he does is he describes that when we say random, we don't mean mathematically random in its purest sense. What we mean is that the mutation isn't the exact mutation that the organism needs in that moment. And the example he gives is this: heat-induced mutations do not produce phenotypic change related to heat tolerance. So if you take bacteria or something and put them in an environment that's too hot for them, that will induce mutations. They will start having more mutations more frequently. And so you might think, oh, well, it's trying to mutate. No, what's happening is that it's not able to maintain its genome because the heat is causing stress on its DNA. Things are breaking and it's having trouble fixing them. The mutations that occur are random, and that they're not specific mutations to help that bacteria deal with heat. They're just changes. It might happen that one of them does help the bacteria deal with heat, in which case that is called mutational rescue. But the bacteria doesn't somehow know what type of mutation it needs to deal better with the heat. It's just experiencing mutations. There is a small chance that one of those mutations will help the bacteria deal with heat, but it's a pretty small chance. A lot of people get a little bit weird when you try to say that cells do things on purpose. And I suppose I'm a little bit less comfortable with that as well. I would say that maybe you could say that cells do things, quote, on purpose, but little quotes around it, because cells don't have thinking minds, so far as we know.
I disagree with his idea of mutations being caused by cells not being able to maintain their genome. I would argue at least some instances of increased rates of mutations are directly related to the stressors in an organism's environment. I agree that cells don't know the exact change needed to deal with an environmental stressor. That is why the rate of mutation is increased rather than mutating once. It seems like there is a tendency towards mutations that could help given the high number of possible mutations and the frequency seen in some noticeable examples of organisms rapidly evolving beneficial mutations. This video shows a recent discovery that might point to the validity of my proposed mechanism.
We're going to be discussing some of the new discoveries and some of the more unexpected discoveries when it comes to the idea of evolution. And specifically, these two studies that seem to suggest that the evolution itself doesn't seem to be as random as we originally thought. But for the purposes of this particular study, or this idea, the scientists focused on just one of the variants. The variant that's usually referred to as HBS mutation, which normally, and surprisingly, protects people against malaria. And the mutation in this particular protein is very interesting because if it goes wrong, it can sometimes produce what's known as sickle cell anemia, which leads to the red blood cells assuming a different shape and not being able to absorb as much oxygen, which obviously creates a lot of problems in the human body. But when the protein creates the HBS mutation, it actually prevents the malaria parasite from being able to infect these cells, basically protecting people in the regions where malaria is a problem. And you might be aware that malaria, even today, is still the deadliest parasite on the planet. The estimates suggest that it kills anywhere from 1 to several million people per year. So obviously having some kind of a beneficial protection from malaria would obviously benefit everyone living in these regions. And this map here sort of shows you some of the mutations and some of the locations of these mutations in regards to the distribution across the world. So for example, you're going to find some of them in India, you're going to find some of the HBS genes in parts of Middle East, also parts of Europe, but a large part of it is in Central Africa. But the thing is, using the idea of random mutations, you would expect these genes to develop everywhere, sort of in all locations on the planet. And so obviously through the process of mutation and through generations of people living in the region, you would expect higher levels of this gene present there, simply because anyone not having the gene is just not going to make it, not going to survive. While at the same time you would expect these genes to also be evolving in, for example, Europe, but not becoming prominent simply because there is no malaria present and no selective pressure. With the basic idea here being that the mutations should still be happening at the same rate. They should still be sort of more or less the same, no matter where people live. Yet this new study discovered something different. They've discovered that people living in those African regions also seem to have much more frequent mutations with a higher rate of generation of that particular protein known as HBS. In other words, the protein mutation that leads to protection from malaria seems to be higher in those African regions. Or to rephrase this, the mutation seems to be generated preferentially in the regions where malaria is present. Something that would be extremely difficult to explain using typical Darwinian theories. And for the study, the scientists actually focused on the idea of the rate of mutation or the frequency of mutation in certain parts of the gene. They didn't really look at how many genes accumulated over time. And they've discovered that this particular gene mutates a lot more frequently in people in those regions as opposed to people living in Europe. With the paper itself providing a little bit more information about how all of this was done and how they used the DNA from various donors to determine that the mutation rate was different depending on where you live. And today it's believed that because of the way malaria works, it's sort of been causing this selective pressure on humans for at least 10,000 years, with certain beneficial mutations producing certain types of proteins, eventually balancing out with the ones that were not beneficial and produce problems. And that's sort of how we usually explain the idea of having certain genes a lot more likely to occur in certain regions. But the frequency of mutation, that's not something that can be explained very easily. Once again, just like with those plants we discussed previously, it means that there is some kind of a bias, selective bias, that eventually develops the frequency that's much higher, even though the species itself is the same, which once again confirms that mutations might not be as random as we initially thought for many years, with the rates of mutation very likely being influenced by various proteins, various structures of chromosomes, potentially the pressure from the environment itself.
This is a case showing a possible link between environmental stressors and the frequency of relevant mutations. There are some other potential examples of these amazing interactions between organisms and their environment. My favorite examples involve obligate symbiosis. Obligate symbiosis is a type of symbiotic relationship where both organisms involved are dependent on each other for survival. Neither organism can live without the other. You can find examples of this and other interesting examples in my book, Purposeful Evolution. Link to the book is in the description below. Thanks for watching, and please subscribe and share the video with anyone who might be interested.